So these three panels are the important ones. Okay, so this is showing you obviously animals, a human animal. We're looking at male and female at ovary and testes. That's the site where meiosis is going to occur. And notice the sexes are separate, right? So we have one sex that's going to produce eggs and one that's going to produce sperm. And no, there aren't any people that actually produce both. Okay, just get that off the table there. They produce gametes, each produces a gamete. These gametes are going to fuse. That is what we title fertilization. And then this technically is a zygote, and that zygote is going to develop into an embryo and then a fetus. Now, when we look at plants and alternation of generations, and if you want to see more detailed pictures of this, I would advise you to look in later chapters, like in the chapter 30, 29, 30-ish. They have some more specific pictures for specific plants for alternation of generations. So what that means, alternation of generations, is in plants, in some plants, as well as some fungus, we have almost the plant lives two lives. It lives one life as a completely haploid, and it only reproduces or makes more cells or more structure by mitosis. So it growth and development here by mitosis. And this looks like a little moss. Okay, so this moss is very tiny. You would be able to see it with a microscope, or actually if you just looked carefully wherever you find moss under a tree or in a nice damp place. So by mitosis, the organism forms a complete organism that looks a little bit different from the alternate generation, and we call one of these the gametophyte, and the gametophyte produces gametes, and we call the other structure the sporophyte, and the sporophyte produces spores. Now, this is going to seem a little backwards to you because gametes mean sex cells, but in this case, they are forming sex cells. They can only make either, you can think about it as plus cells or minus cells. We don't usually refer to them as male and female because particularly in these plants, they both have flagella. So they're both small and flagellated. And they're microscopic, right? Just like your gametes are. I guess that's more common in fungus. They're both flagellated and protists. In this particular case with the moss, one of them actually makes a structure like this, and the other produces little cells that swim. And they're separate. So there's a male gametophyte plant and a female gametophyte plant, and they're both haploid, and they can only make N gametes. When those gametes find each other, usually in a drop of water, they're going to fuse and form a zygote that's going to become 2N. And then this portion here, a new structure comes on the top of the gametophyte. That's the sporophyte structure that's made by mitotic division. So we have lots of cells and we make this little cap here. And inside the cap, that's where meiosis is going to occur. And meiosis produces the spores that are N, either male or female, to produce male or female gametophytes. I find it's confusing. I know you're gonna have to look at it, you're gonna have to think about it, you're gonna have to listen to it, you're gonna have to read about it several times to get it straight. I find the easiest thing for me to remember is if I think about the gametophyte produces gametes, so produces cells that are N, the sporophyte produces spores. The gametophyte produces gametes by mitosis. The sporophyte produces spores by meiosis. And that may help you remember and keep it straight. And then I think there's also a protozoan picture here. So this is another example. And in some cases, this zygote, this 2N form, is very, very short-lived. Right? In this case, there's no sporophyte that's produced. It's just a zygote that immediately undergoes meiosis to form spores, basically, or little N cells that are then going to form an organism that's going to make more gametes. Oh, I like that picture. There's a good picture of the crossing over. And yes, you need to know the vocabulary for the crossing over as well. I didn't mention it during our discussion, but the actual proteins that hold the sister chromatids or the tetrad together, that's called the synaptonemal complex. And then you can actually see the crossing over that's happened here only in prophase 1, capital highlight. And the marks of where there has been a crossing over, that's what the chiasmata is. So if we take a look back here in this picture of all of the steps of meiosis, you can see in prophase 1, they're labeled the chiasmata, that's the plural. I believe chiasma is the singular, and that marks the place where crossing over has occurred. And then notice those chromosomes that have crossed over, those pieces that have broken off and changed, they keep them color-wise all the way through. So you can see here's a chromosome, a blue chromosome that has a piece of red chromosome 1 on it, and it's exchanged, and this red one has 2 pieces of blue on it. The longer the chromosome, the more likely it will cross over, and the more times that there will be, the number of crossovers increases. With really short chromosomes, not as much crossing over. Okay. Stop the share. And then somebody asked a question. I think you do. If you don't, I'll post it. I thought I put it up there as well.